The overall goal of this procedure is to identify, isolate, and clone human T cells with a defined alloantigen specificity. This is accomplished by first isolating T lymphocytes from whole peripheral blood by density gradient centrifugation. In the second step, the alloantigen-specific T cells are labeled with fluorophore-conjugated peptide HLA tetramers and enriched by magnetic separation. Next, the labeled T cells are isolated by single-cell flow cytometry cell sorting. Finally, the single-cell cultures are stimulated with anti-CD3 and anti-CD25 antibodies. Ultimately, the alloreactivity of the antigen-specific T cells can be evaluated by flow cytometric analysis.
The main advantage of this technique over other techniques such as bulk lymphocyte stimulation and limiting dilution is that this technique enables the generation of T cell clones with specific reactivity to well-defined alloantigens.
Generally, individuals who are new to this method may have difficulty in culturing and expansion of the T cell clones. This requires careful daily monitoring of the cultures.
Before beginning the procedure, aliquot 4 milliliters of room temperature density gradient into 2 to 4 sterile 15 milliliter conical centrifuge tubes. Use 70% ethanol to wipe the outside of 1 to 2 filled green top blood collection tubes containing blood from a healthy volunteer, and then carefully remove the tops. Decant the blood into a sterile 50 milliliter centrifuge tube and then